Conjugation is one of three mechanisms that bacteria use to conduct lateral or horizontal gene transfer and thus become genetic recombinants. Conjugation is different than the other two mechanisms, transformation and transduction, respectively, in two critical ways. First of all, conjugation is considered to be a direct form of gene transfer. And the other way that conjugation is different is that it is purposeful. So let me elaborate a little bit more on those concepts. Here we have a picture of conjugation between a donor cell that is packaging up some DNA and sending it through this conjugation pellis to the recipient cell. So DNA goes directly from the donor to the recipient, and that's what we mean by direct. If we look at transformation, there's an intermediate step in that transfer of DNA. The donor cell actually dies and releases its DNA into the environment, and then that DNA is picked up by a recipient cell. Again, what I just described is transformation, which was covered in the previous lecture. Likewise, transduction is indirect because it uses an intermediate step that involves a virus. So conjugation is really the only direct mechanism by which bacteria can transfer DNA from one cell to another. And because it is a direct transfer, where both parties are willing participants, both the donor and the recipient, it is also purposeful. If we think about transformation, the donor cell has to die first before that DNA is released. So obviously the donor isn't really a willing participant in that. The bacterial donor is not purposefully giving up its DNA to a recipient. And transduction is a similar thing where the donor is not purposefully giving its DNA to a recipient. So conjugation is different from the other two mechanisms in that it is both direct and purposeful. We are going to be looking at conjugation in E. coli, in part because it is perhaps the most well-studied bacterial species for conjugation. Here I have 3 different E. coli cells. You'll notice that the two on the left have a blue chromosome and a pink plasmid. In the top left picture, the plasmid is separate. In the bottom left picture, the plasmid seems to have fused or is at least sitting next to the blue chromosome. The right cell, which has a green chromosome, has no plasmid. So there are many different types of plasmids, but the plasmid that we are going to look at is called the F plasmid. The F plasmid is so named because it contains the F factor. The F factor is a gene, and it's going to be one of several genes on this plasmid. The F factor enables that cell to create a conjugation pellis. So we're going to go ahead and put a little mark there and an F to denote that the F factor is one of the genes on that plasmid. Now, if a cell has an F plasmid, we call that an F plus cell. If a cell does not have an F plasmid, we would denote that as F minus. Now, one of the interesting things about plasmids is that they can actually fuse with the chromosome. So what we have here is a picture showing this plasmid really close up with that chromosome. It's still an F plus cell in this iteration. But what's going to happen is the DNA is actually going to fuse together to create one larger chromosome where the plasmid DNA is incorporated in with the chromosomal DNA. So the F factor is still going to be embedded in there, but it's no longer really an F plus cell because there is not an F plasmid. I mean, there is an F plasmid, but it's not in a plasmid form, which would be a separate piece of DNA. It's fused with the chromosome. So what do we call this? Because the reality is that these three stages can pretty readily go back and forth. So we need a way to describe a cell where the F plasmid has fused with the chromosome. It's not really an F minus cell because it could easily re-extract its plasmid. So we're going to give it a different name. And the name that we give this one, where the plasmid has fused with the chromosome, is HFR. And HFR, it stands for high frequency recombinant. You don't need to know that, but that's what it stands for. So basically what we have here are three different iterations. We have an F plus cell, and an F plus cell is one that has a true and distinct F plasmid, but the F plasmid can fuse with the chromosome. And when that happens, we call it an HFR cell. Or we can have a cell that does not have an F plasmid in any way, shape, or form, and we call that an F minus cell. Now, E. coli conjugation has a couple interesting rules associated with it. One rule is that conjugation is a one-way street. In other words, DNA flows from a donor to a recipient. It is not a double exchange. The recipient doesn't give something back. And the donor is always going to be the one that carries the F factor. In other words, the donor must make the conjugation pilus to initiate conjugation. Another bizarre rule, but it seems to be a rule nonetheless, is that the recipient must not have the F factor. So if we go back up here to our iterations, we have F plus, HFR, and F minus. We can map those onto whether or not they would be a donor or a recipient based on our rules. So the donor must carry the F factor. And the only two iterations that carry the F factor are the F plus cell, or its counterpart, the HFR version of the F plus cell. So the donor is either going to be an F plus cell or an HFR cell. And if it is true that the recipient cannot carry the F factor, then the recipient must always be an F minus cell. So we basically have two different permutations of this conjugation. We could have an F plus conjugating and donating DNA to an F minus, or we could have HFR conjugating and donating something to an F minus. So those are the only two possibilities for this E. coli conjugation model. So what we're going to do is we're first going to take a second look at how these two go back and forth, and then we are going to talk about when a cell does this, what is the outcome, and when a cell does that, what is the outcome. This is your textbook's image showing how an F plus cell, which is over here, can become an HFR cell. And what's not really shown is that we can go backwards too. So an F plus and an HFR cell are interchangeable, meaning that these forms of how you carry the F plasmid. Are you a distinct F plasmid? That would be an F plus cell. Or is your F plasmid fused with your chromosome? That's going to be your HFR cell. So now that we've taken a second look at how F plus and HFR go back and forth, let's take a look at conjugation between an F plus and an F minus cell. On the left, I have an F plus cell. And on the right, I have an F minus cell. So this is going to be conjugation of F plus to F minus. Remember that DNA always goes from the donor to the recipient. And the donor must be F plus or HFR. Okay. There's my little F factor. So essentially what happens is the donor cell, the F plus cell, is going to start synthesizing a conjugation pilus. So let's go ahead and draw that in real quick. To create a tunnel between these two cells. And then it's going to make a copy of its plasmid. So that donor cell is going to make a copy of its F plasmid, and that F plasmid is going to use that conjugation pilus for direct access to the recipient. And then eventually that conjugation pilus is going to break. The conjugation pilus is pretty delicate, so it doesn't take very much force to snap it. Okay, so an F plus to F minus conjugation specifically transfers the F plasmid. And note that the recipient is no longer going to be F minus, because the recipient now has their own copy of the F plasmid and therefore is F plus. You might ask yourself, what is the point of this? If you're just transferring around the F plasmid from one cell to another so that they all become F pluses, then what? They can all make a conjugation pilus, but if there is no F minus cell to do further conjugation, it seems pointless. What's not being stated here is that the F plasmid can carry other useful genes as well, such as genes for antibiotic resistance. And this is what we saw in Lab 14, where the F plasmid not only carried the F factor, but also a gene for ampicillin resistance, which confers a big benefit to that bacterial cell, especially if that bacterium is a pathogen inside of a human body and the human is taking antibiotics. So now that we have looked at F plus to F minus conjugation, where again the plasmid is transferred, let's take a look at the textbook's image of this. So you'll see that we have an F plus cell as the donor, and F minus cell as the recipient. The F factor is going to be copied and sent over to the recipient, and the recipient becomes F plus. So conjugation where you transfer an F plasmid is always F plus to F minus. So now let's take a look at the other version of conjugation that I outlined, HFR to F minus. So here we have an HFR cell and an F minus cell. And once again, the donor is going to be the HFR cell because they carry that F factor, which allows them to synthesize a conjugation pilus directly to the intended recipient, which is F minus. But now there is no F plasmid to transfer. So what is going to be transferred? Well, it turns out that part of the chromosome is going to be transferred. Every genome has what is called an origin of replication. So I'll put the origin of replication there. And then you start copying one strand of that DNA in either a clockwise or counterclockwise fashion. So here, what's going to be copied is a little bit of the F plasmid and quite a bit of the chromosome. And then that DNA is going to make its way over to here. And then through the steps that we showed in a previous lecture, only part of that DNA is going to recombine with this F-. So for instance, let's say that the F- cell decides to keep just the part in yellow. So after a fashion, what we would expect is something like this. So once it takes just that little bit of blue, then it's going to go ahead and cut up all of the extra stuff that it's not incorporating, like so. And that is now a recombinant. Now notice that the F- cell is still an F- cell. It is a recombinant F- cell, but it is not an HFR cell, and it is not an F+ cell. So why is it that this recipient is not an F+ cell? It does not have a distinct F plasmid, so therefore it is not F+. But wait, isn't it HFR? No, it's not. Why not? An HFR cell must be able to extract a complete F plasmid. And while there is some new DNA that is brought over, you can't extract that and make an F+ plasmid. There's no F factor in those genes. So therefore, it is neither F+ nor HFR. It is simply a recombinant F-. So here's the textbook's image of this particular process. So we start with an HFR cell, that's the donor. The F- is a recipient. You can see that it's sending some of its DNA over in the middle picture, and then some of the new DNA is kept right there in the recipient. So a really important takeaway here is that conjugation can involve either the transfer of a plasmid or the transfer of part of a chromosome. However, the stage that the donor cell is in will determine what is transferred. If the donor cell is in its F+ state, then it transfers over a plasmid. If the donor cell is in its HFR state, then it transfers over its chromosome. Now, one thing that I kind of glossed over was, why did only a certain amount of the chromosome get transferred? Why not the whole thing? Well, if conjugation was allowed to proceed for up to 2 hours between an HFR cell and an F- cell, then the entire chromosome might have been copied and sent over. So the goal is always to do the entire chromosome and transfer it, but because the conjugation pelus is very delicate, and we'll just show another quick image of that here, so you can see just how delicate it is, the conjugation pelus will often break before the entire chromosome is transferred over. So how long the conjugation pelus is allowed to stay intact for determines how much of the chromosome gets transferred. The longer the conjugation pelus is intact, the more of the chromosome is transferred over. All right, that concludes our lecture. Thanks.